So this is an outline of the talk. The first thing is that we are going to talk a little bit about preparing your data for voxel-based group analysis, although I think pretty much everything that you learned last week was in this section of preparing the data. So we're just going to review. And then the second thing is to talk about what your dependent variable is going to be in your group analysis. So you will have hopefully have a good sense, especially with the lectures that Tom gave on the general linear model, as well as the stuff last week about how you would get a single number per voxel per person. And it's that numbers, those numbers that are going to go into our group level analysis. So then we're going to talk about pretty simple single sample analysis. So what you do if you have one group of subjects and you like to say overall, where did they activate in the brain? And then two sample analyses where you have two groups or one group at two occasions and you're trying to compare the activation between those two occasions, two samples. And then we'll go to a little bit more tricky situation, which is multiple factor and group level regression analyses. And then even more tricky is sort of the idea of looking at sort of the spatial patterning and multivariate analysis and classification algorithms, which I really consider a little bit more advanced group analysis. So I'm just going to touch the surface of that and not go into great detail because I think it's enough to figure out how to do a t-test for and some of these more simple group analyses. Then I'm going to talk a little bit about the idea of effect size and effect size maps, and sort of in general about how to present, how best to present the data, that the results that you get in group analysis, and then talk about region of interest analyses. So the preparation is first of all, you want to have already done an analysis of your individual level data. Way back in the day, when I first started doing FMRI, people would use each person's observation as almost as an independent observation, and they were doing what's called sort of mixed effects analyses. So they were, at each time point that the person has was being counted as an N. So they would publish papers that would say, you know, I have an N of 8,000 or something like that. So because they had, you know, 1,000 observations on 8 people. And unfortunately it took a little while for people to say, hey, those are not really, that's fine, you can publish that paper, but if you do, you can only generalize to those 8 people. And what most people want to do is generalize to all the people in the sample from which you drew a population from which you drew those 8 people sampling. Therefore, you need to, in general, what people do now is they analyze each individual's data using a general linear model and then come up with a variable for each person in each voxel. And then part of that may be these things that you learned a little bit about last week: smoothing, co-registration into of the functional to the anatomical to make sure it's aligned, normalization into some kind of common atlas space so that everybody is in the general same location in space so that you can put them together. Mask out, a lot of people do some things taking out the extracranial signal because we don't really expect, unless there's some kind of weird ESP thing that we don't know about, they don't really expect there to be significantly active voxels outside the brain on a group level. And maybe if you had a slice coverage that was not the entire brain, you might want to restrict it to the voxels that all the subjects have data. So if you have a group analysis, that way you know that everybody had data in that voxel. You don't have some places where only 8 people had data and other places where all 12 people had data. And then you probably talked a little bit about motion and the importance of looking at motion and correcting for motion. And you may, before you go into a group analysis, also want to just flat out exclude some people. So if they had, you know, our general rule of thumb is if more than a third of the time points in a given run were found to have high levels of motion, whatever that cutoff might be, then you might just say, you know, that person's kind of a loss. There's just not enough information about their time course to include them in the group analysis. But if you do that, you going to want to be sure that you're not excluding more people in, say, your experimental group than in your control group, or in your sort of pre versus your post, or things like that, so that it's that you're not biasing your analyses because you had to exclude more people from one group than the other. And then if you have multiple runs of the same task and the same condition, then you're going to need to, before you hit group analysis, you're going to need to decide how you're going to deal with that. So as you'll see, when we get to some of the software packages, there are ways of incorporating multiple runs per person in a repeated measures kind of design, so you can do it at the level of the group analysis. Some people, if they have those runs where just like back to back to back, they may actually concatenate the runs together into one long run, and then just do a time series analysis, a GLM, on an individual subject basis, so you get that whole thing. Some people might analyze each individual run and then average the output from that in each voxel across the 3 runs as kind of like a more reliable measure of the response to the task across time. So that's just something based on your hypotheses that you'll have to decide. So what is the dependent variable is an important question that you need to answer before you start your group analysis. For example, if you are looking at an event-related design, so can anybody just briefly say what the difference between an event-related and a block design is? Yeah, and so specifically what would be different in an event-related versus block in terms of how the stimulus was presented?
The block you presented in equal kind of like blocks. And then there'll be like either in between each block, then you have a resting, in between those two, and then event related is a little bit more random.
Right, and there would just be sort of like one trial or one stimulus and then some time to let the response to that die down, and then another one. Yeah. So in an event-related design, you might be wanting to look at the hemodynamic response or the bold response at different latencies after the stimulus occurs. And so you might be using some tent function or other ways to get a to get a single number. Your goal is to try to get a single number in each voxel for that particular condition that you gave. And so that number may be a beta weight from a GLM where you had a block of where it was on versus off, or houses versus neutral, or faces versus houses, or whatever the contrast it is. So that's another question, is which contrast are you trying to analyze? And this totally depends on your hypotheses, right? You need to figure out which thing are you want to contrast, because as I'm sure you've already heard multiple times, bold fMRI, you don't just get a single number for a single condition. It's always a contrast of how much more bold signal did you get in this situation than in this other situation. So you need to figure out which contrast you're going to analyze in your group analysis. And you may have looked at a bunch of different ones. So let's say, for example, you had an end-back working memory task where people saw letters flashing on the screen, and one condition, you were just supposed to press a button whenever you saw the letter X. That's sometimes called a 0-back condition. Or a 1-back condition would be that you have to press a letter press the button if the letter that you saw is the same as the one you just saw. Run 1 before and then 2 back, so measure working memory. And so you have to decide ahead of time whether you're going to contrast 2-back versus 1-back, 2-back versus 0-back, 1-back versus 0-back, or all those things. So you may have calculated all those things, but for your group analysis, you need to be really specific about what your hypothesis is so that you know which contrast to put into your second level or group analysis. And there's been some data out there showing that the effect sizes of fMRI, and this may not be a big surprise to you, the effect sizes in fMRI are pretty small. I mean, even for something as sort of sledgehammery as having somebody like press a button repeatedly. In the motor cortex, we're still talking about pretty small effect sizes of like the order of like 0.8 or so for a Cohen's d. So the contrast that you pick for your group analysis is important. You want to pick the one that has the most contrast if you can, because doing something that's like the brain activation of, you know, houses versus faces is going to be harder to, you know, they're going to have a less reliable contrast there than if it's houses versus nothing, for example. Okay. The other thing to think about is that in each voxel, you actually have multiple values that come out of your GLM, right? You've got the fit coefficient, you've got the t statistic, you have sometimes a p-value associated with that t statistic, and a lot of times people have calculated a percent signal change from some baseline level of activation. So you have a bunch of, in AFNI parlance, a bunch of sub-bricks at every voxel, right? And so again, you have to decide which of those sub-bricks, which of those numbers are you going to put into your group analysis. In general, it's not really recommended to use the inferential statistic, so a T or an F or an R if you're doing some kind of a regression type analysis, because the distributional properties of those test statistics are not ideal for being then to go into a group analysis. So just like any kind of statistical test, you have to look at the assumptions of the statistical tests and what the assumptions are about the distribution of the variables that go into it. And so the dependent variable needs to be, if you're using these linear models, most of which I'm going to talk about are all linear models, you want that dependent variable to be nicely normally distributed. Percent signal change is nice because it's very interpretable. You can sort of say this is how much the signal went up when this kind of stimulus came on, and it's a really good thing to show in graphs and in even to paint on the surface of the brain, that kind of thing. But it's not necessarily the greatest thing to put in as your dependent variable in group analyses, because percent signal change can only go from 0 to 100, and so that truncated thing, it's just not usually doesn't usually have a great distribution. It's often skewed. And so the one that I have tended to use and that many people use is the fit coefficient or the parameter estimate or the beta weight in a GLM for the contrast of interest. Yeah.
If you're going to use non-parametric statistics with the percent signal change for justice, like do people do that, or is it just easier, or are there other reasons that people decide to use something like this?
I think that it could be fine for non-parametric tests. It's just that there's not as many options for non-parametric tests in the stats world of neuroimaging, and so it makes it just a little bit more challenging to do the kind of models that you want to do if you want to do it with non-parametric statistics. So I think people tend to want to use that, but then that might not be the case if you're doing a region of interest analysis, for example, and you get the average percent signal change in there, and then you can throw it into your favorite stats program and do a Mann-Whitney U test between groups, and you're fine. So yeah. Okay. So any questions about this? So we've done our group, GLM, our individual level analysis, which by the way, I think that Alan is going to spend more time on Wednesday. So if you guys feel like you're not quite there yet in terms of how to get the individual subject level, I think he's going to go back to talking about that and specifically how to do it in AFNI with 3D deconvolve. So, there will be more on that, but basically you do your individual subject level analysis, and you basically have a brain map of, so you have a 3D spatial array of one number per voxel per person, and it's whatever contrast you've picked and whatever dependent variable like a fit coefficient that you've picked. So then now what do you want to do with that? So again, this is going to depend on your hypotheses and what you're interested in analyzing. And here I like to say that if you ignore the fact that we have like 20,000 or 30,000 voxels, and we just zero in on, zoom in to look at just one voxel, then all of this is no different from your stats class for any other thing. It could be the average accuracy on a cognitive task. It could be reaction time. It could be number of cells that you counted in a given brain region, or whatever it is that it is. It's just boiled down to one dependent variable. And now all of this stuff is really just ways of doing massively univariate testing of your hypotheses. So hopefully, if you are somebody who's recently taken a stats class, none of this is going to seem too unfamiliar. But what I'm going to just try to point out is how these types of tests are implemented in AFNI, which is the program that I'm most familiar with. So for a single sample analysis, which is actually something that maybe we don't do as much as normal, like normally we wouldn't usually say just like, is the accuracy of this group of people greater than 50% or something like that, but sometimes you might. But in neuroimaging, we do it a lot, because a lot of studies, for example, in cognitive neuroscience, are just taking a big group of otherwise healthy, usually college students, and asking the question, like, where in the brain does this task activate? And so for that, all you really need is a single sample analysis, because you're asking sort of where are there voxels that responded with a greater response than 0 to this contrast. Or to the stimulus. And so the simplest approach actually for this would be to just take all your folks that are in the same standard space, like say Talarac or MNI space, and then just use a 3D calc or some kind of calculator to calculate the average fit coefficient across all those folks. But just like you just the mean can be pulled by outliers, and there's not really any test, then there's no test statistic that goes along with that. So you can get a nice picture of the average activation, but you can't really say how reliable that effect is. And so that's why people use the single sample t-test. And when you do that, then you end up with multiple subbricks. In general, you get an overlay or a first subbrick or a 0th subbrick that is the mean intensity. So really it's just that. It's just averaged across all the people in each of the groups, and or across that single group that you had. And then a t-test, a t-value that's given to you to assess the reliability of the effect. And the null hypothesis is that whatever your dependent variable in that voxel is 0. And it just does that same t-test 20,000 times. And then, gives you the ability to paint the color of the average intensity on there and threshold it based on the t-values.
20,000 times you mean because it's doing it per voxel?
Yep.
Okay, just
yeah. So we're gonna get, next week, Alan's gonna talk about clustering and thresholding. And obviously that's a big deal. That's one of the biggest differences between doing this on, 8 cognitive tests versus 20,000 voxels, is you have a huge chance of false positives, right? So we have to figure out ways of making sure that the activations that we see are reliable. And we can, in contrast to now not ignoring the fact that this is a brain where there's a spatial location to each of these voxels, is the idea that that people have come up with is that it would be one thing if there was a bunch of really strongly activated voxels all scattered around the brain, but if you see a bunch of them all right next to each other, then that makes you a little bit more certain that maybe this is an area of the brain that is activated. And so this spatial clustering and thresholding is usually used in combination to figure out what are the most reliable findings from a given analysis. But for now, I'm going to just be talking about how you get a value of the test statistic in every single voxel, and a little bit about effect sizes, and then you'll learn later about how to protect against all those false positives and get a sense of what is the most reliable finding from your group analysis. So the program in AFNI that is recommended now to do this is called 3D Test++. It's one of the most straightforward group analysis programs that exist. And I think it's pretty funny that it has plus plus. I mean, so now, somebody decided that was a good idea to name it that way. And then, forever we have to type those extra pluses. Like, why not just 3D test, not 3D test plus plus? Anyway, and there was a 3D test that wasn't as good as this one. But I don't know why two pluses. So anyway, so the first thing that you have to do in your command line, and in your assignment, which is off sort of by a week, so the assignment for this is going to come out sometime next week. But in your assignment, I have the link to the help file, but you can look on AFNI and look at the help file for 3D Test++, and it gives you lots of information, more than you ever really want. But basically, you have to specify which sub-brick for each person. So you probably have a multi-brick file for each person that has like different contrasts in there, and the intercept, and some t-values and some fit coefficient values and things like that. So you have to say which one of those you want to be used for the t-test. And so that usually is something like this, where you're specifying sub-brick number 3, which in this case I've decided is the contrast of houses versus fixation. I just made this up. And then if you only enter one set, so the syntax is set A and set B, because t-test is only for 2 groups. So set A and set B. So if you only enter set A, then it will know that you want to do a single sample t-test. You also can include a covariate if you'd like. So for example, if you'd like to see whether it's significantly greater than 0 activation in a voxel controlling for the relationship of age to the bold signal in that contrast, then you can add age as a covariate. If you would like, by putting in a covariates file. And then what you get is the mean fit coefficient across all the participants and a t-value, where the degrees of freedom on the t-value for the slider, it's going to tell you the p. The p-value for the t-value is based on degrees of freedom of n-1, where n is the number of participants in the group. So this goes back to, again, that you get, it's 1 value per person, so you get 1 degree of freedom per person. And yeah, so this is an example of the syntax. You would just do 3D t-test++, set A, and then you would have subject 1, subject 2, all the way up to subject number 13. And you would say, I want to call this, and this is just an example of how I sometimes name my output files. I might put like healthy controls, HC underscore n equals 13 to remind me how many people went into this analysis, underscore houses, because that's the house sub-brick that I was looking at here. Any questions about that?
So you have to add each individual subject of the command line. Is there a way to like have some sort of text file where you have the list?
I don't think that there's a way to have a text file in 3D t-test++. However, I did see something the other night when I was looking at the help file that suggested that there was a way to use wildcards. So for example, if you had like subject 1 underscore house, subject 2 underscore house, subject 3, that I think there is a way that you can make it a little bit easier if the file names are nicely regular. I haven't done it myself. Generally what I do is I just make I do an ls, or I list the files, and then I just copy and paste them into a editor, text editor, and then search and replace for the bracket 3 and do it like that. There may be easier ways to do it, but that's kind of how I usually do it. And just in general, when I do my group analyses, I almost always have some kind of a text editor that I'm working in, and I either make it into a batch file that I can then run again, or I just cut and paste from that text editor onto the command line. But it's nice to have a record of the analysis that you did, so that you can, if you need to redo it or whatever. So it's kind of like in SPSS having like a syntax file or something. Any other questions about single sample analysis? Okay, so then if you're going to get a little fancier, you might want to compare two groups. So this might be two different groups of individuals, like patients versus healthy comparisons, males versus females. And one of the things that, another mistake that people made in the early days of fMRI, which I thought was interesting, is that they would, for example, they might say, okay, I'm going to examine the effects of biological sex, so I'm going to look at the difference between males and females. And what they would do is they would show a picture of the brain activation of males and say these are the areas that were significantly activated, and then they would show another picture of the brain activation of females and say these were the areas that were significantly activated. And then they would say, see how these are different areas? This area is, much more active in the females, and then you don't even see that in the males. And they wouldn't actually statistically test whether there was a difference in that degree of activation for males and females. So that's obviously a no-no. And so this type of program will allow you to do that direct statistical comparison. You can certainly still show a picture of the single sample t-test for males and a picture of the single sample t-test for females, but then you're also going to want to show a picture of the direct contrast between them. And we'll get back to why that's so important when we start talking about effect sizes. And the other way that you can use a two-sample comparison is, of course, if you have the same individual but at two different states or two different conditions. So on drug versus off drug, before or after some kind of a training program, sleep deprived versus well-rested, or even task A versus task B. So if you had people in the scanner and they were doing one task that was really difficult, and then they did a similar task that was really easy, and you want to look at the effect of difficulty, you can contrast those two tasks. If you have like a run that has like a 0 back, a 1 back, and a 2 back, though, I would recommend that you not use group level analysis to do that, because although you might get sort of the same answer, you might as well do your individual level GLMs and do all the kind of controls for nuisance of regressors and everything to compare your conditions that way, and then put that contrast into this. But this is more if you have like two completely separate runs that you want to contrast two conditions. And then the null hypothesis in this case, of course, is the mean of group A is different from the mean of B, or sample A is different from B. And you would use the same program, 3D t-test++, and if they're independent samples, then you don't have to do anything special. If they are, if it's a within subject design and it's the same people but just in different conditions or pre or post, then you should specify the -paired option so that people know, so that the program knows that those folks go together. And you have to, again, specify the subbricks that you want to use. And, it might be two different subbricks of the same person, but generally it's two different things. Be aware of which is set A and which is set B. So you have to just say set A is the sample and set B is the sample. And the reason why it's important to be aware of it is because what they show you is, what the default is that it's going to show you the mean, the mean of set A minus the mean of set B as your overlay. So the color scheme is going to be A minus B. So for example, if you were doing something that was like pre and post training, I would recommend that you make the post training set A and the pre-training set B, even though it seems a little counterintuitive, because then, I don't know, I just think it makes more sense to have things be red that got bigger in, you know, a bigger response in the post after training compared to before training. So but make sure you really keep track of this, because this throws people off all the time. They make their pictures, then they're like, wait, what does red mean again? Oh, right. You know, and similarly, people make different decisions. Sometimes people will, if you think that your, say, healthy comparison group should have more activation than your other group, pathological group, then you might make healthy controls be set A and the other group set B, so that, again, red areas are where healthy controls are more active. But you might do the opposite, just keep track. And then you have to, again, specify the subbricks. You can include a covariate, again, but here you need to be really careful with 3D t-test++ in terms of choosing whether or not to center your covariate. And I'm not going to go into detail about that because you probably heard about this in your statistics class, but think about where you would center your covariates, and also watch out if your covariate means are very different between the two groups. So, for example, if you were going to test people with schizophrenia versus people without schizophrenia, and some kind of measure of like nicotine dependence as your covariate, it might not necessarily work very well, because you're going to have a lot of nicotine dependence in the schizophrenia group that you don't have in the healthy control group, and so it's going to be difficult to control for that statistically when there's such differences in your covariate between groups. And again, that's just a general statistical principle, so be careful with that. There's discussion of both of these things in the help file for 3D t-test++, which is like 10 pages long or something. Okay, and then what you're going to get out, again, is two subbricks. One is going to be the mean of set A minus set B. So that's going to, again, be red for areas where set A is greater than set B, and it's going to be blue for areas where set B is greater than set A. And then you're going to have a t-statistic that is with the degrees of freedom of n of A plus n of B minus 2. And then the method of calculating variance differs in the independent and paired, why it's important to put that in there if the observations are paired, because they may be correlated. So this is an example of how you actually write this out, 3D t-test++. You would have set A, and then you would have, for example, like I said, like a post-test value for every subject up until subject M, and then you would have set B would be a pre-test for all those same subjects. It would be the same subbrick because it's houses in this case, and then you would say paired because each person had a pre and a post, and then a prefix might be something like healthy controls, n equals 13, houses, post minus pre. And I like to, again, just put what minus what, like A minus B right there in the title so that I don't forget which way I did it. Again, you can look at it in AFNI in the history, you can see what was run, but it's just nice when you're just looking through your list of files to be able to see that. And then another example that's like more of a between subject one would be 3D t-test++, where set A are the healthy controls and set B are the bipolar disorder subjects, and the prefix might be HC n equals 13, BD n equals 13 houses. Okay, any questions about that? All right, so moving right along. So what if you have more than two groups, or more than two samples, or more than two factors of any sort, like different conditions or difficulty levels or things like that? Then you still may be interested in lots of different things. You might be interested in between subject differences, so there might be 3 different diagnostic categories, maybe bipolar disorder, major depression, and healthy control. There could be within subject things, so 3 different task conditions, or 4 different things where there's multiple pre and post manipulations. There might be sort of random effects that are representative of a larger population, like the subject, or fixed effects with different levels where the different levels are intrinsic interest, like gender, for example, or set doses of a drug. And all of these factors could have 2 or more levels. And so you want something where you might be able to combine all these things together. And so then you would be able to test main effects, interactions, and plan contrasts looking at particular factor level means. So again, this is just the usual, this is sort of general linear model, ANOVA, ANCOVA, mixed linear, linear mixed effects, etc. And luckily for us, there's a couple of different ways that you can do that in AFNI. Kind of unluckily, for whatever reason, I think for efficiency, time efficiency, these programs run in R. So you have to make sure that your AFNI package that you also downloaded R with that, and you have all the R libraries that are supposed to go with AFNI. Because this one does run in R. And it also means that you have to be familiar enough with RStudio or using R to be able to put the commands into R as opposed to putting them into Linux command line type thing, or Windows command. So anyway, 3D MVM, which I think is multivariate modeling, is what that stands for. So it's similar to the independent samples t-test, but it's when there's more than 2 levels or factors. And so again, there's a good help file that you can take a look at, and in terms of how you specify the different sub-bricks and which ones go with different factors. You don't have to do any dummy coding for this, which is nice. So you can just say like males, females, you don't have to create dummy codes. And you can specify follow-up contrasts and then sub-contrasts. And you can have missing values, which is nice. Earlier versions of ANOVA in AFNI did not allow you to have you had to have exactly the same cell size, which was really annoying because it wasn't even like missing values. Like if you just designed your experiment where you had like 12, or you're not even designing your experiment, but you ended up having 12 healthy controls and 16 people with bipolar disorder, you couldn't run that ANOVA because you had to have the exact same cell size. So this is nice because you can have missing values. Although the recommendation is that if the missing values are in a within subject quantitative value, so something that changes, that's paired within subject, but it is some kind of a quantitative as opposed to a dummy coding type thing or discrete variable, then you should use the next program that I'm going to talk about, which is 3D LME. So 3D linear mixed effects also runs in R. It's a bit more flexible, but it's also a little bit more complicated. So probably as you're getting started, unless you know that you have that situation or something, somebody said on the AFNI message board that you should really use 3D LME, I would just go for 3D MVM because it seems a little bit more straightforward to use. Or again, if you have something where you really feel like you have some nested random and fixed effects that you need to take care of, and you would, if it wasn't brain imaging, you would use a linear mixed effects model, then you should use that one. So here's an example for 3D MVM. I'm going to hit some highlights, not walk through the whole thing. But basically, this is the output prefix. I think 4 jobs, I think this has to do with running things in parallel. And then these are the between subject variables. So in this case, this example, there was genotype and sex. And then scanner was, so these were things that they wanted to test effects of, and this was kind of more of a nuisance variable. So that's why it gets a plus instead of a star. And then within subject variables, condition and emotion. Sum of squares type 2, number of contrasts is 14. And so then they go on to actually talk about all the different contrasts and specify what they want. So for example, here's an interesting one, the sex by condition interaction. So testing whether there's differential effects of the condition in males versus females. And so they code that to say that 1 is male and -1 is female, and 1 is face and -1 is house. So you have to specify that, and then you'll get out things. And then here there is like a data table that you enter in here And this specifies the subject number, the genotype, the sex, the scanner, the condition, the emotion, and then which input file goes along with all that unique combination of things. Yes.
So I'm just trying to like figure out the relationship between this and ours. So it's like you're basically just like entering all the information into the command line that R would then use, and it just sends it to R and says do this. Okay.
Yeah. So and yeah. So it's basically, instead of writing it in the same, like C is what AFNI is a C program. So instead of writing in C, basically they wrote it in R. So you can't just throw it on the command line and expect it to run. There is no AFNI program called 3D MVM. It's an R program, an R script basically. It's called 3D MVM that you're invoking. And then if you want to do group multiple regression, so for example, if you want to identify the relationship between brain response and some combination of discrete and continuous variables, including something like how's age related to the magnitude of brain response to faces, or what parts of the brain are most related to mean accuracy on the cognitive task that they were doing, then these, and that whatever that continuous variable is at the level of subjects, as opposed to like at the level varies within a subject within a run, then you can also use 3D LME. So it's very flexible in terms of mixing discrete and quantitative variables and allowing you to look at that. Okay, so first in-class survey for today. So those of you who are doing the Canvas thing, I put everything on the slides too, so if for some reason you're not in Canvas, but if you can find the group analysis 1 in-class survey 1, hopefully that's obvious. I think I published it correct. Is it there? Under quizzes. Okay. So for all of the questions, this is the setup. Consider this study design. You test 10 people with major depression and 10 healthy comparison participants on 2 different days, once following a normal night of sleep and once following a night of total sleep deprivation. The FMRI task is an in-back working memory task with 3 levels of difficulty: 0 back, 1 back, and 2 back. For each individual, you have calculated the contrast of 2 back versus 0 back using a GLM. You have also measured sleepiness levels just before scanning in all participants on each day of testing, but some test sheets got lost, so data are missing at random. I know, the RA needs to be fired, right? No, I'm just kidding. Okay, so the first question is, which AFNI program would you use to see which areas are most activated by a high working memory load, meaning the 2 back, in healthy comparison participants after a normal night of sleep? You guys are welcome to chat if you want to figure out what you think the answer is. And then, Tom, is there like a way that I can look at?
Yeah, there's survey statistics. I'll pull it up. I'm not sure if you set it up as 4 questions.
Yeah, 4 questions. Oh, I didn't say what the right answer was, though.
Yeah, well,
I guess I'm supposed to say that.
Yeah, it'll assume the first one's
Oh, it does?
yeah,
Okay. Okay, so we won't look at the.
okay, so just, yeah.
I'll just start with. Okay.
So I can tell you how what the once the statistics show up, I can show you what I'll just
Tell you what people answered. Okay, thank you. But by doing it this way, I did do it so it's not graded. It's graded to take it 1 if they participate. Right? Yeah.
From looking at the relationship to age, for example, single sample, can you use 3D test processes to use ages? Doesn't matter who.
Yes, it will produce the relationship with the covariate. So yeah, you could use that.
I mean,
if it's just a single sample,
yeah.
and yeah. All right. So what's the consensus? It's our highest answer.
We can't submit it because there's 4 questions we have.
You just submit all 4 questions.
Yeah.
What?
What you said.
Okay.
Submit all 4 questions.
Yeah.
Okay, sorry, I'm a novice on this. I had no idea what I was doing. I know you offered to help me too, and I just didn't take you up on it. I'm like, I can figure it out. Sorry. Okay. Okay. So we'll again, it's not like graded anyway. So does anybody want to say what they think the answer is? The third one, 3D t-test plus with all healthy participants as set A. Anybody want to pick another one? Or is everybody happy with that? Okay, yeah, so this is a pretty straightforward thing. You've already done the GLM to get the high memory load contrast, and that's a single group and on in a single condition, and so you're just interested in this one thing, so that would just be a single sample t-test. All right, so next. Which AFNI program would you use to see which areas differ in their activation to high working memory load depending on whether the previous night had normal sleep or total deprivation in the depressed group? Okay, which one are people liking this time? What's that?
The third one.
The third one. Okay. Good. Yeah. So the first one would be comparing depressed and healthy comparisons. That's like your sort of normal 2-sample t-test. The second one would actually probably lead to the exact same answer as the 3D t-test because it's the MVM with a between subjects group, which would be depressed versus healthy comparison. So that should give you the exact same answer. And the last one is a little more looking at kind of an interaction between the things, but we were only looking in the depressed group at a within subject. So you need that paired option, and then you would just put their normal night versus their total sleep deprivation night in there. Day after, right? Okay. And then question 3, which AFNI program would you use to see in which area brain response during high working memory load is more sensitive to sleep deprivation in depressed patients than healthy comparison? Total sleep deprivation. All right, which one do we like on this one? What? What's that?
Oh, we know the second.
The second one, yes. Yeah, so I kind of gave this away when I was explaining the last one, but yeah, so this is, there'd be a lot of other stuff, but the most important thing would be that there would be a contrast here where you're looking at the interaction of sleep, which is the either normal or total sleep deprivation, with group, which is either healthy control or depressed, or healthy comparison or comparison, because this is the way that I said it maybe wasn't super clear, but basically more sensitive to sleep deprivation, I meant sort of that there's a bigger difference between rested versus deprived in the depressed than there is in the healthy comparison. So that's an interaction effect they're looking for. And you would have you would have made it clear that the group variable was a between subjects and the sleep variable was a within subjects. So it's kind of a mixed model there. And yeah, the rest of them are not. Last one, what AFNI program would you use to identify the areas of brain response to high working memory load that are more sensitive to sleep deprivation in depressed patients while controlling for sleepiness levels? So this is that sleep questionnaire that we gave. Right. So which one do we like for this? The first one? Okay. That one is good, but it also has the, it's contrasting the two groups, so it's an interaction with group, whereas in this case, I was kind of going for just, well, but so actually that one would probably work, but if you remember some small fact that I told you, when there's missing data, then you prefer the 3D LME. Because it deals with that, and specifically when there's missing data for a within subject quantitative variable. That's why I added that little thing about the lost sheets of paper.
And that's all you have to do for indicating that you want sleepiness as a like control variable.
So I think that you just there's going to be more that you will have to do in terms of specifying the contrasts, which I didn't include here. But this is just the top part, that you want sleepiness to be in there and in the model, and that.
But there's no like additional options. It's not in the contrast, but just additional options.
No, I don't think so. Okay. All right. So all of these methods that we just talked about, as I mentioned before, are kind of massively univariate. So they assume that you have activation levels for each person at each voxel, and then they just do the same thing at every single voxel. But there are other group analyses that more explicitly incorporate the spatial nature of the data to look at kind of group patterns of activations or networks, so to speak. Now we're going to be talking about functional connectivity, which is one way of looking at networks, but this is really more in looking to see kind of do the same places. This is a very colloquial way of saying it. Do the same places light up within a group, and does another group have another pattern of places that lit up? Okay, where you're not really getting at that if you're just saying in this group these are the places that lit up above a certain threshold, and in this other group these are the places that lit up, but you're not directly comparing whether the pattern, the spatial patterning of the activation response is different or the same in the two groups, because you're only testing at each voxel, and no voxel really knows anything about the analysis is agnostic to where that voxel is in space. But there are things like partial least squares, independent components analyses, and then more machine learning type things, support vector machines that can help to look at the patterns of response across different groups and compare those responses to do, say, classification analysis, for example. This is just an example of a partial least squares analysis where they have looked at they found this, they found a pattern that is correlated with, that changes with load. So they had 3 different loads. And this pattern is sensitive to load, and it's sensitive to load in a way that differs between the groups. And then they have another pattern that's sensitive to load in a different way, and it's more similar across groups. So usually what you do is you end up showing pictures of the pattern that you found, and then going back down and showing how it's related to whatever it is that it was related to, and how it's different between the groups in your sample. But it's a little tricky in terms of how you do statistics in terms of comparing the pattern between groups. Because you have to decide what is the normal pattern, right? So if you're trying to say like the people with schizophrenia have a different pattern than the healthy controls, you could say the healthy control pattern is the norm and the schizophrenia are different from that. Or you could combine the 2 groups to say that's the normal pattern, but these people are different from that overall pattern, taking into account all the variability between the groups. Or you could have a separate comparison group. And it's just very difficult to statistically compare patterns between things. So it's hard to find something that's not just a form of voxel-based or ROI analysis, but classification algorithms can do that. And there's a lot of interest in using these sort of machine learning, right, to classify groups based on their pattern of brain activity. Another thing that this has been used for that you might be familiar with is sort of mind reading tasks, where basically you see if you have the algorithm try and find the pattern of brain activity that predicts what the person was seeing. And then you can see if you apply that algorithm to other people, then you can guess what they were seeing, which is kind of cool. Okay. But another thing to keep in mind about these is that, I've talked a lot about T statistics, and you're going to learn a lot about, or T or F come out of these programs. And you're going to learn a lot about the clustering methods. That's these cluster threshold methods where you're trying to find the peaks of activation, right? But it's also important to remember that right underneath those peaks are the bottom part of the mountain. Right, so it's just the peak is not just like floating up there and there's nothing right next to it. There's a gradual, the mountain may be steeper or wider, but there's going to be a gradual thing, and all those voxels are kind of really close to being significantly activated, but they just didn't quite make it. And so that's why it's really important to show, I think, the effect size maps, so that you can see exactly what's underneath the peaks. So yes, show the peaks, but also you may want to show everything. So this is an example of different thresholds that are being added here. And this is a paper that I have on the assignment as well, Jernigan et al. And actually Chris Fenneman-Ostein, who was here, was one of the co-authors of this paper, saying that it's really important if you, for example, if you were just to look at the top here, you might say that this tapping task only activates the right motor cortex. But then if you look down here, you can see that, or sort of that area near the insula there, you can see that yes, there is that hot spot here, but it's not like there's nothing going on over here. And probably if you did a direct comparison between the area, this activation and this activation, it may not be statistically significantly different. It's a little bit lower in magnitude, but it's not that it's absent there, and yet it makes it look like it's absent when we do this thresholding that we do. So it's really important to keep in mind these effect sizes, and that's why it's important to always do a direct test. But even when you're interpreting your results, a lot of people, when they interpret the results, they're like, these were where the peaks were, and there was no activation in the other places. That's where you get in trouble, because it's like there was no significant activation there, but you don't know that there might have been something just below your threshold. Okay, and so this brings us to the final topic for today, which is region of interest analysis. So region of interest analysis, we've been talking about voxel-based analysis, where we're doing all of these statistical tests in every single part of the brain. And those are really nice because they allow us to see all the possible places that there could be a response and all the possible places where there could be differences between groups. But as we'll see, there's a lot of downside to it in terms of the statistical power, because of the fact that we have to correct for all these multiple comparisons and the high likelihood of false positives with that kind of thing. And so oftentimes, not always, but you may actually have a strong a priori regional hypothesis based on lesion literature, based on previous MRI or FMRI studies. You may be really mostly interested in how your task, which differs from the task that's been out there in the past, is related to activity in this region, or how the two groups are different in that particular region. And so, if so, you'll probably want to apply a region of interest or an ROI mask. But also, region of interest analyses can be helpful to understand your voxel-based analyses. And understand what exactly is going on in those clusters that you've identified as being different between two groups. And I'll get back to that a little bit more. There's different types of region of interest. One general class is anatomical ROIs, meaning regions that are defined based on the brain anatomy as opposed to the brain function. So those anatomical ROIs could be individual subject ROIs, meaning that in each person you identify a region either by hand tracing, say, the hippocampus based on atlases and guidelines about where the hippocampus starts and stops and how to distinguish between the white and gray matter, and you may do some painstaking tracings of the hippocampus or the amygdala or something like that. Or you might apply a software package such as FreeSurfer, which uses automated methods as well as probabilistic atlases to determine for each subject where their hippocampus is, or where their amygdala is, or where their dorsolateral prefrontal cortex, although it wouldn't be called that in FreeSurfer, but where their particular regions are in cortical or subcortical areas, and then use that to apply to see how much functional MRI activity is there in that person, and then put that into a group level analysis. Or you could apply your anatomical ROIs at the group level, so you could use atlas-based. So once you have everybody in a standard space like MNI space or Talarex space, then you could take those atlases and the divisions, like say Brodmann's area 9, and make a region of interest, and then apply that to the group effects, or apply it back to each individual subject in the normalized space, and look at that as well. And then a final sort of quasi anatomical ROI is meta-analysis based. So these days there's been a lot of effort to put together the results of fMRI experiments in different domains. So for example, language domain, you can go online and you can say to Neurosynth, and you can say, give me all the regions that are involved in language comprehension. And it will give you a map of language comprehension regions based on meta-analyses of many different papers out in the literature. And then you could use that map as to apply to your own data and see what is that group differences within, say, the language region. And then functional ROIs are where you define your region of interest based on how the fMRI, the where the peaks of the fMRI signal are. And so you could take those clusters that you've done from your current study, or you could take clusters that you found on another scan, another person's scan previously, and apply those and try to see what's going on in those regions that were found to be the most different between groups or the most related to age or something like that. We're going to talk about some caveats to that. But then another thing is if you had, for example, a peak of activation, or somebody they didn't do a meta-analysis, but some other person had done like the exact same task you did, and they found that the strongest activation was in this particular spot within the anterior cingulate cortex, then you could create yourself a little spherical ball ROI right around that peak, equidistant on all sides from that coordinate that was seen in the other study. So there's different ways of actually making these ROI masks, because basically what you need in order to get information out about what's going on in a particular sub part of the brain is you need a mask, and a mask is just a three-dimensional brain mask, a three-dimensional brain map that has zeros everywhere except for ones in the region that you want to find out about. Or if your mask has multiple different regions, AFNI really likes it if you have like ones in the biggest region and then all twos in the voxels of the second biggest region, then all threes. And so having that 1, 2, 3 allows you to get a nice printout that separates the values and the statistics in each of the different ROIs. Does that make sense? So you need to make these ROI masks. And so there's different ways of doing it. So in the Andy's Brain blog has a really nice video about making masks, which I also have the link to in the assignment. You can use the graphical user interface of AFNI to call up atlas-based things. So you can say, I want Brodmann's area 9, and it will paint it on whatever anatomical underlay you have, and then you can say, fill that with ones, and then for the left, and then you can say, I want you to fill the right BA9 with twos, and then save that out, and then you'll have yourself a mask that's zeros everywhere and ones in the left and twos in the right, Brodmann's area 9. And then he goes through how you can use this program 3D Undump to create a spherical ROI around a coordinate. So you can see how that works, or you can do the help file for 3D Undump. But if you have a coordinate or where you want, you have to also know the orientation of your data set, like whether it's in RAI or LS, LPI, etc., in order to get it to work correctly. But you can create a nice spherical ROI and then look at it in AFNI and make sure that it's in the right spot. And then if you have a threshold, if you want to use your own data to make a mask of all the places where you had activation, and you have like a clustered map, so you have like 3 different clusters of activation that were significant, then you can use this 3D merge -1 cluster order option to get them to turn that into 1s, 2s, and 3s in each of those to make like a nice mask out of that. Questions about this? I know it's sort of glossing over the surface, but it's one of those things where it would take a really long time to like go through every example, but we'll probably be doing this in the practical, so you'll get some experience then. Okay, but one of the things you really need to watch out for with any of your region of interest analysis is this idea of non-independent analyses or circular analyses. So if I did an analysis where, for example, I looked for the voxels in which there was a strong relationship between age and brain activation to the 2 back versus the 0 back condition, and then I found clusters that were highly related to, where the brain response was highly related to age, and then I used that cluster as a region of interest and either picked out the fit coefficient from the highest voxel in there, or I averaged all the fit coefficients in that cluster, and then I plotted that versus age in the same group, then that's completely circular. Like, it's going to be a really high correlation because that's where I got the cluster from, is all the voxels that had a really high correlation. And so obviously it's going to have a really high correlation. So what you need to make sure of is that you're not sort of basically patting yourself on the back and being like, wow, look at this really high correlation that I got in this region, because obviously you did, because that's how you found the region to begin with. Now, if I had said, I think there's going to be a high correlation with age in the hippocampus, and I traced the hippocampus, and then I averaged the fit coefficients within the hippocampus, and then did a scatter plot of the hippocampus versus age, then that would be totally fine. That's not circular. That's just, I had an a priori hypothesis about where, how that would be related to age. It's when you use the relationship to find the voxels and then show what the relationship is within those voxels that you can get really inflated regions. So the other thing that this article pointed out is that a lot of people were reporting correlations between, say, brain and behavior that were in like the 0.8, 0.9 range for correlations, which seemed very unlikely given the fact that both the brain measure and the behavioral measure probably didn't have reliabilities that high. And you may know that you can't have, the correlation between two variables cannot be higher than either of the variables' reliability, test reliability. So how correlated they are to themselves, they can't be more correlated to something else than they are to themselves, if that makes sense. So there was some problems with the literature, and people were doing this kind of double dipping or non-independence, or what they called voodoo correlations. And so it's important to watch out for that kind of analysis and not do it. On the other hand, I feel like in some ways they did a little bit of a disservice to the field, because now people are so afraid of these kinds of follow-up analyses that they're not doing them in order to better understand the significant result that you got. And so I'm going to give you an example of that. It's really hard to understand because with fMRI we have all these different contrasts, so we have like 2 back versus 0 back, and then we have depressed versus healthy controls, and then we've got pre-sleep deprivation versus not sleep deprivation, and you get a picture of a cluster and it's red, and you're like, that's a significant interaction. How do you understand the nature of that interaction without plotting it out, right? That's what you would do with anything. You would probably do some kind of a scatter plot and see where the crossover is in the lines if there's a significant interaction. So you need to have some follow-up analyses of these clusters, and it's just important that you're not say that that's some kind of new finding. You're just saying, now I'm following up on this finding that I had to understand it and to understand the magnitude of that effect. So let's say that you have your region of interest, like the boundaries of a cluster that you found to be significant, or the boundaries of some kind of anatomical area, either in an atlas or a hand-drawn or whatever. What can you do to summarize the data within a region of interest? So one of the things you can do is you can count the number of voxels within that region that exceed a certain threshold. This is sometimes done because, especially if it's kind of a big region and there might be some voxels that are negative or deactivations or opposite, a negative contrast, and some that are positive. And so you might say, I'm just going to count the number that are positive because that's the direction of the contrast that I'm interested in. So I'm just going to count how many voxels were activated. But the tough thing about this is really hard to figure out what the appropriate threshold is, because now we're talking about in an individual subject and understanding, like, what t-value would actually make sense. The distributional properties of those counts are not that great. So this is generally not recommended. Poldrack says don't do it. I have done it in the past, but probably better is to do something like averaging the fit coefficients within that region and then using that. Apparently SPM uses the first principle component of the voxels that are in your ROI with the idea that that's sort of like the main driver of that ROI's similarity, I guess, the similarity of the voxels within the ROI. And then you might also actually do a voxel-wise analysis within an ROI. That's sometimes called a search region of interest, or sometimes called small volume correction. But you might say, I don't want to average across all the voxels in this ROI because maybe it's sort of big, like maybe it's like the whole superior temporal gyrus or something like that. And so you're like, I think I'm only interested in effects that are in this gyrus and not everywhere else in the brain. So I'm going to give myself a bonus in terms of power and not have to work so hard to correct for false alarms if I just look at this small part of the brain. So that you might also just do it voxel-wise and not collapse at all within that. But then once you have your ROI stats, your statistics that you got out of your ROI, then you can do whatever you want with them. Take them into your favorite graphing program, take them into your favorite statistical analysis package, and do things like make scatter plots of the relationship between brain activation in that region and some other behavioral measure. You can make dot plots to show each individual data point in a given region in one group compared to the other. I'm going to show you some bar graphs, but I actually think that bar graph, like, why not use a dot plot where you have an individual dot for each person so that you can really see the variability and see, whether your means being drawn by some outliers, see the overlap in the distributions. Just generally is much better if you have individual dots for each subject in your neuroimaging analysis. And usually, unless you're using like archival data or you're doing Enigma work or something like that, you're only talking like 50 people at the most. So it's not like it's hard to plot each one. And yeah, you can do any kind of analysis you want. So that, like going back to the beginning, the question about non-parametric analysis, you can definitely do any kind of non-parametric or any kind of analysis that you'd like. So here's the way that you can get out the information that's within an ROI in AFNI. You can, 3D mask-ab is the most straightforward way to just get a simple average of all the voxels that are in a mask. But if you want to have more options, you can use 3D ROI stats. And like I said, if you have more than one blob in your mask and each of them has a different number inside them, then 3D ROI stats will give it to you in different rows of like a CSV file that you can bring into Excel. And it also has some other nice options, like for example, you can tell it to take the average but only over non-zero voxels. So for example, if you have an ROI that might be like on the edge of the brain and some people it may go off the edge of the brain, then you can just get it to give you the average of the non-zero voxels. You can also get standard deviation, some other nice things from 3D ROI stats. But just keep in mind that you can't use 3D ROI stats with your mask data set and your individual subject GLM data set if those two are not at the same resolution. So if you can imagine, it's just basically taking the mask and laying it down on top of the voxels of your FMRI, and if those voxels are different sizes, then it's not going to be able to figure out how to calculate the mean within the ROI. So you may have to do downsampling with a program called 3D Fractionize, because usually your mask is at a higher resolution because it's often based off an anatomical. So sometimes, if there's a mismatch, it's usually that the mask is too granular, it's too high resolution, and so you have to make it to be less high resolution like your FMRI data are. Okay, this is an example. Maybe somebody turn the lights down. This is an example of some ROI stats kind of thing. So this is from the autism lab that I work in. Teresa knows about this. So these are typically developing kids, language delay, ASD, autism spectrum disorders who have good versus poor language. And so this is the NeuroSense meta-analysis areas that are supposed to be involved in language. And so this is not applying this ROI, but just showing a comparison. So this is thresholded, but not masked in any way. And so you can see that, for example, the ASD4 just really don't have a lot of activation in the sort of canonical language areas. This is all during sleep. Let me do the scans here at the center. And then this is the contrast of all groups versus ASD4, and so you can see the ASD4 have a lot less activation here. But then applying these frontal and temporal regions of interest to these same data sets, you can see very clearly that it's the ASD4 that have very different activation, particularly in the temporal regions, whereas the ASD who have good language actually are looking a lot more like the typically developing kids. And the other thing you can see here, one of the reasons why it's good to plot what's going on, is you can see that in this right hemisphere language ROI, the ASD actually have a large negative signal change, meaning that they have sort of more activation to the contrast here, which is just non-speech, than to speech. Okay, so then this is the final thing, is the last in-class survey. So this is a scatter plot from a study of mine of amygdala activation during a facial emotion processing task. It's right and left, or the difference between the X's and the dots. And the y-axis is the beta weight for the contrast of negative faces versus oval shapes. So it's a face identification task. And then x-axis shows the z-scores for an affective empathy score that they did outside the scanner. So this was during a face task, not during an empathy task. And this was an affective empathy task that was done outside the scanner. And so the question is, oops, I don't know what happened to the question. Do you guys see the question? Okay, so can somebody read the question out? How did I get that in here?
Which of the following best describes the results shown in the figure? This is a voodoo correlation with circular non-independent analysis. People with high affective empathy show a stronger amygdala response to negative faces than to ovals. C, the amygdala of people with low affective empathy is insensitive to negative emotion. Or D, the people with low affective empathy have a stronger amygdala response to negative faces than ovals, and the amygdala of people with above average affective empathy responds slightly more to ovals than to negative faces.
So I probably pretty much gave it away with the fact that that last answer was really long. But I think my point was just that, just because you see this negative correlation, it's really important to see where the zero lines falls, which is why I put the lines at 0 here. So what you can see is that most people... is that it's really, the correlation is being driven a lot by these people that have low levels of affective empathy, and they're the ones that are really reacting, their amygdala is really reacting to the negative faces, whereas the people who have higher levels of affective empathy are kind of near 0, so they don't have a big difference in their amygdala response to the faces. A little bit for these ones over here, but in general, it's important to be able to look at these kind of scatter plots to understand how to interpret them, because where they are based on 0. So this is just a couple. These were the amygdala ROIs that I made, and you can see that they're resampled at the level of the FMRI. So what's next is, like I said, Alan's going to go over GLM for individual subject data and the use of 3D deconvolve. He's going to talk about clustering, why you have to do that, how you do it, and a little bit more about how to present and interpret your FMRI group data. So, and be looking next week for the assignment, and the practical will be next Friday, the 14th, for this topic. Because this Friday is last week's practical, basically. It's like off by a week now. Thanks, everybody.